Did you know that DNA or deoxyribonucleic acid in all human beings is 99.9% identical? It is that 1/10 of 1% difference that makes us all unique. DNA derives its name from deoxyribonucleic acid, a type of nucleic acid. Nucleic acids are made up of polynucleotide chains, which are formed by several nucleotides or molecules that make up the structure of the DNA when bonded together. In fact, the length of a DNA is defined by the number of nucleotides or pairs of nucleotides present in the DNA. A pair of nucleotides is also known as a base pair. For example, E. coli has 4.6 multiplied by 10 raised to the power 6 base pairs, and bacteriophage lambda has 48,502 base pairs, while the haploid content of human DNA has 3.3 multiplied by 10 raised to the power 9 base pairs. It was in the year 1869 that Friedrich Miescher first identified DNA as an acidic material present in the nucleus and called it the nuclein. However, technical limitations made it very difficult to isolate such a long polymer intact, and therefore no further interpretations regarding the structure of DNA were made for several years. Finally, in 1953, James Watson and Francis Crick proposed a simple double helix model for the structure of DNA. They did this with the help from the X-ray diffraction data that was created by Maurice Wilkins and Rosalind Franklin. Further, James Watson and Francis Crick proposed that base pairing existed between two strands of polynucleotide chains, which was a distinctive attribute of their proposition. This proposition was based on the observations made by Erwin Chargaff, who found that the ratios between adenine and thymine and guanine and cytosine are constant and equal to 1 for a double-stranded DNA. In fact, due to base pairing, the polynucleotide chains possess a very unique property, that is, the strands of the base pairs are complementary to each other. Therefore, if we know the sequence of bases on one strand, it is possible to predict the sequence in the other. Moreover, when two strands of a parental DNA separate, each serves as a template for synthesis of a new daughter strand because of complementary base pairing, and the two double-stranded daughter DNA generated are identical to the parent DNA molecule. Thus, these discoveries provided a clearer picture to the genetic implications of the DNA structure, and soon the DNA double helix structure and its simplicity in explaining genetic implications became revolutionary. Let us now learn about the chemical structure of a polynucleotide chain present in the DNA double helix structure. The polynucleotide chain is made up of three components: a nitrogenous base, a phosphate group, and a pentose sugar, which is deoxyribose. Further, the nitrogenous base is of two types, purines and pyrimidines. The purines comprise adenine and guanine, while the pyrimidines comprise cytosine and thymine. Now, through an N-glycosidic linkage, a nitrogenous base is linked to the pentose sugar, forming a nucleoside, which can be guanosine or deoxyguanosine, adenosine or deoxyadenosine, uridine or deoxythymidine, and cytidine or deoxycytidine. Conversely, depending on the sugar present, a nucleotide or a deoxynucleotide is formed when a phosphate group is linked to 5'-OH of a nucleoside through the phosphoester linkage. Also, a dinucleotide is formed when two nucleotides are linked through a 3', 5' phosphodiester linkage, and several such nucleotides can join similarly to form a polynucleotide chain. At one end of this chain is a free phosphate moiety at the 5' end of ribose sugar, which is called the 5' end of the polynucleotide chain. Similarly, at the other end of the chain is present a ribose with a free 3' OH group, which is called the 3' end of the polynucleotide chain. A salient feature of the double helix DNA structure is that it comprises two polynucleotide chains, the backbone of which is constituted by sugar phosphate with bases projecting inside. The double helix structure has several other salient features too. Its two polynucleotide chains have anti-parallel polarity. That is, if one chain has a polarity 5-3-, the other has 3-5-. Moreover, the bases of the two DNA strands are paired with the help of hydrogen bonds forming base pairs. Adenine is bonded with thymine from the opposite strand with two hydrogen bonds and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bonds. Because of this structure, a purine always comes opposite to a pyrimidine, which results in a uniform distance being maintained between the two helix strands. Another salient feature of the helix structure is that the two chains are coiled in a right-handed fashion. Therefore, the pitch of the helix is 3.4 nanometers, and each turn consists of about 10 base pairs. As a result, the distance between a base pair and a helix is around 0.34 nanometers. Additionally, in a DNA double helix, the plane of one base pair stacks over the other, which, along with the hydrogen bonds, makes the helical structure very stable. The stable DNA helix structure allows it to be the genetic material that has instructions for the development and functioning of all known living organisms. Did you know that the length of a DNA or deoxyribonucleic acid double helix in a typical mammalian cell is about 2.2 meters? This length is obtained by multiplying the total number of base pairs present in the DNA double helix with the distance between two consecutive base pairs. Now, the total number of base pairs present in a DNA double helix in a typical mammalian cell is 6.6 multiplied by 10 raised to the power 9, and the distance between two consecutive base pairs is 0.34 nanometers, or 0.34 multiplied by 10 raised to the power minus 9 meters. By multiplying these two factors, we get the length of the DNA double helix as 2.2 meters. Have you wondered how such a long polymer is packaged within a typical nucleus with a dimension of about 10 raised to the power minus 6 meters? To understand this, let us learn about the packaging of a DNA double helix, which is different for both prokaryotes and eukaryotes. In prokaryotes such as E. coli, the nucleus is not very well defined. However, the negatively charged DNA is not scattered throughout the cell, but is arranged in large loops, is held together by a few positively charged proteins in a region known as the nucleoid. On the other hand, in eukaryotes, the manner in which a DNA double helix is packaged within the cell is more complex. Eukaryotes contain a single molecule of negatively charged DNA packaged around a spool of positively charged basic proteins known as histones. The charge in a protein is decided by the abundance of amino acid residues with charged side chains. Now in this case, the proteins are positively charged as the histones are rich in basic amino acid residues like lysines and arginines, which carry positive charges in their side chains. Furthermore, the histones are arranged to form a unit of 8 molecules known as the histone octamer. Now around the positively charged octamer, a negatively charged DNA molecule is wrapped to form a structure known as the nucleosome, which is held in place by the H1 histone. A typical nucleosome has around 200 base pairs of DNA helix, and the nucleosomes present in chromatin can be seen as a beads-on-string structure when observed under an electron microscope. It is the nucleosomes that make up the repeating unit in chromatin, the thread-like bodies present in nucleus. Now, the chromatin fibers are of two types, euchromatin and heterochromatin. In a typical nucleus, euchromatin fibers of 30 to 80 nanometers in diameter are loosely packed and stain light. On the other hand, heterochromatin fibers of about 300 nanometers in diameter are more densely packed and stain dark. Moreover, of the two types of chromatin fibers, euchromatin is transcriptionally active, while heterochromatin is inactive. These chromatin fibers coil further and condense to form short and thick bodies called chromosomes during the metaphase stage of cell division. In this way, a DNA double helix is packaged into a chromosome, which is further packaged within the nucleus. If we stored the entire DNA sequence of a single human cell in books, we would have 3,300 books, each with 1,000 pages, where every page contains 1,000 letters. Yes, a human genome, which has about 3 multiplied by 10 to the power of 9 base pairs, can produce such an enormous amount of data. In fact, an ambitious international scientific research project called the Human Genome Project, or HGP, set out to do just that, to determine the complete sequence of base pairs of DNA inside the human genome. More precisely, the HGP aimed to identify approximately 20,000 to 25,000 genes in human DNA. The average gene consists of 3,000 bases. However, gene sizes vary greatly. Did you know that dystrophin is the largest known human gene, which has 2.4 million bases? Now, if you add the number of base pairs in the human genome, the sequences of more than 3 billion chemical base pairs would have to be determined under the HGP. Therefore, by aiming to understand the genetic makeup of the human species, the HGP served an important purpose. It was important because it is a sequence of bases in our DNA that determine our genetic information. 99.9% of all nucleotide bases are exactly the same in people. Yet we differ from each other, and that is because our DNA sequences differ in certain places. Scientists have identified about 1.4 million locations with single-base DNA differences, also known as SNPs, or single nucleotide polymorphism. Knowledge about variations in the human genome can equip us with more ways to diagnose, treat, or even prevent disorders and ailments that affect human beings. Similarly, knowledge of the repetitive sequences in DNA shed a lot of light on chromosome structure and evolution. Repetitive sequences, also known as microsatellites, are those stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times, in the human genome. Also, they have no direct coding functions. In fact, less than 2% of genome codes for proteins, and for more than half the discovered genes, are still unknown. With the advent of advanced genetic engineering methods, it was now possible to isolate and clone a piece of DNA to determine its sequence. So even though the HGP was a mammoth project, it was a viable one. Since the HGP aimed to store the entire genome information in databases, it required high-speed computational devices for data storage and retrieval. In addition, the HGP required improved tools for data analysis. Thus, the HGP is also credited with contributing to the rapid development of a new branch of biology called bioinformatics, which involves the application of statistics and computer sciences to the field of molecular biology. The HGP was a 13-year project that began in 1990 and was completed in 2003. It was coordinated by the U.S. Department of Energy and the U.S. National Institute of Health. Did you know that initially it was James D. Watson, one of the co-discoverers of the double helix structure of DNA, who headed the project? However, in the initial years of the HGP, Wellcome Trust of the UK became a major partner, though more contributions arrived from countries such as Japan, France, Germany, and China, to name a few. Although the HGP had aimed to understand the genetic makeup of the human species, it also focused on several other non-human species such as the bacterium E. coli, yeast, a free-living non-pathogenic nematode called Caenorhabditis elegans, a fruit fly called Drosophila, and the laboratory mouse. Learning about non-human DNA sequences can lead to a further understanding of their natural capacities and traits, which may offer solutions in various sectors such as health, agriculture, and energy production. Apart from transferring information and technologies to other sectors, the HGP also intended to address ethical, legal and social issues, or ELSI, that may have developed while working on the project. Therefore, though the HGP was a mammoth and hugely beneficial project, it required enormous effort, cost and innovation as well as caution to accomplish it successfully. In 1869, Friedrich Miescher isolated an acidic material from the nucleus of a cell and called it nuclein, now known as nucleic acids. Also around the same time, Gregor Mendel proposed the principles of inheritance. In fact, Gregor Mendel, Thomas Hunt Morgan, Walter Sutton, and numerous other scientists had narrowed their search for genetic material to chromosomes. However, as chromosomes of eukaryotes contain several proteins along with DNA or deoxyribonucleic acid, scientists couldn't exactly confirm whether the genetic material was DNA or proteins or both. By around 1926, the search for the mechanism of inheritance of genetic material had reached the molecular level, yet the genetic material had still not been discovered. However, in 1928, Frederick Griffith, a British scientist, conducted an experiment that accidentally demonstrated the transformation of genetic material in bacteria. In fact, Frederick Griffith was in the process of developing a vaccine against the bacteria Streptococcus pneumoniae that caused pneumonia by inoculating the dead microbes along similar lines of Louis Pasteur's experiments on vaccination. Frederick grew the bacteria in a culture plate and found that they were of two forms, the smooth and the rough. This was because the smooth shiny colonies or the S strain of the bacteria possessed a mucus or a polysaccharide coat, while the rough colonies or the R strain had no such coat. Further, when the mice were injected with the S strain, they died of pneumonia infection, while those injected with the R strain did not develop pneumonia. This proved that the S strain of bacteria was virulent or lethal, while the R strain was harmless. Moreover, Frederick also found that heating killed the bacteria. So when he injected heat-killed S strain bacteria into the mice, they did not die. On the other hand, when he injected a mixture of R strain bacteria and heat-killed S strain bacteria, the mice died. Surprisingly, he even recovered the living S strain of bacteria from the dead mice. So on the basis of this experiment, Frederick concluded that the heat-killed S strain bacteria had somehow transformed the R strain bacteria. He also concluded that the reason why the R strain bacteria produced a smooth polysaccharide coat and became lethal or virulent was because of the transfer of some genetic material from the heat-killed S strain bacteria. However, there was still no means to ascertain or identify the genetic material that was transferred and responsible for the transformation process. Later, after 16 years, in 1944, a team of 3 scientists, Oswald Avery, Colin MacLeod, and Macklin McCarty, once again began research on the experiment that was once conducted by Frederick. At that time, they tried to identify the biochemical nature of the transforming principle in Frederick's experiment. For this, the scientists first purified biochemicals such as proteins, DNA, and RNA or ribonucleic acid from the heat-killed S strain bacteria. They did so to identify the biochemical responsible for transforming the live R strain into S strain bacterial cells. Through their experiments, they discovered that it was the DNA from the S strain bacteria that had transformed the R strain bacteria. They also found that digestion of the DNA of heat-killed S strain with DNase inhibited transformation. Further, the scientists also discovered that addition of proteases or protein-digesting enzymes and RNases or RNA-digesting enzymes did not affect the transformation. This led to the conclusion that proteins or RNA were not transforming substances and implied that DNA was the cause for the transformation. Thus, through their experiments, Oswald Avery, Colin MacLeod, and Macklin McCarty concluded that DNA is the hereditary material in most living organisms. Nevertheless, many biologists were still unconvinced that DNA is the hereditary material. and so the search for genetic material continued for several years and was concluded with the discovery of DNA. Once it was proved that DNA was the primary genetic material in the living system, it was natural to conclude that DNA replicates as a molecule, qualifies as genetic material only if it replicates. Scientists Watson and Crick, who in 1953 had proved that DNA had a double helical structure, had also suggested a probable scheme for the replication of DNA. According to their proposed scheme, the two strands separated and acted as a template for the synthesis of two new complementary strands. After the replication process was completed, each DNA molecule would end up with one parental strand and one newly synthesized strand. That is why this DNA replication scheme was referred to as semi-conservative. In 1958, to find conclusive evidence for this scheme of semi-conservative DNA replication, experiments were conducted by scientists Matthew Meselson and Franklin Stahl on bacteria Escherichia coli or E. coli, a bacteria that divides every 20 minutes. Meselson and Stahl grew E. coli in a medium that had NH4Cl15 isotope as the only nitrogen source. N15 isotope is a heavy isotope of nitrogen, but it is not radioactive. Many generations of E. coli were bred in the same medium, which was not too hard to achieve since a new E. coli generation arrived every 20 minutes. As a result, newly synthesized DNA too had the N15 isotope as a constituent of purines and pyrimidines. This new heavy DNA molecule was distinguished from normal DNA by virtue of its high density while being extracted via centrifugation in a cesium chloride or CsCl density gradient. Later, E. coli cells were transferred into a medium with normal NH4Cl14 isotope. After the cells multiplied, their DNA was extracted and separated independently in CsCl gradients to record the density of DNA. It turned out that DNA extracted from the first progeny of E. coli after the transfer from the N15 isotope to the N14 isotope medium had an intermediate density. It was referred to as hybrid DNA. The next generation of E. coli, which was born after 40 minutes, had equal amounts of hybrid DNA and light DNA. Thus, the findings of the Meselson and Stahl experiment established the validity of Watson and Crick's theory of the semi-conservative DNA replication scheme. Similar experiments were further conducted in higher organisms such as plants and human cells to prove the same. One such experiment was conducted by Dr. J. Herbert Taylor and his colleagues in 1958. In root tip cells of Vicia faba or faba beans, using radioactive thymidine to determine the distribution of newly synthesized DNA in chromosomes. Here again, the experiment proved that DNA replication even in chromosomes was semi-conservative. Therefore, with multiple experiments conducted by different scientists, it was concluded that DNA replicates semi-conservatively. DNA replication is a fundamental process that all living organisms undergo to copy their DNA.